we're fortunate to be able to hear from some of our newest faculty from each of our constituent faculty divisions. We will start with Bowen Li, who will be joining the Faculty of Pharmacy in January 2022 as an assistant professor. He will be launching a cutting-edge research program focusing on biomaterials, immuno-engineering, and mRNA delivery. Bowen will then be followed by Helen Tran, who is an assistant professor in the Department of Chemistry in the Faculty of Arts and Science. Her research program leverages the rich palette of polymer chemistry to design new materials encoded with information for self-assembly, degradability, and electronic transport to address challenges in health and sustainability. Following Helen, we'll hear from Rafael Montenegro Burke, who is an assistant professor in the Department of Molecular Genetics in the Temerty Faculty of Medicine. His team focuses on the development of strategies for novel metabolite characterization and discovery to expand our view of conserved metabolic pathways and their impact on cell function in health and disease. And closing out our faculty presentations is Christopher Lawson, an assistant professor in the Department of Chemical Engineering and Applied Chemistry in the Faculty of Applied Science and Engineering. His lab works at the interface of engineering and biology using innovative approaches for systems and synthetic biology and bioengineering to engineer microbial communities to recover valuable resources from wastes. And with that, I will stop sharing and allow Bowen to start with his presentation.
Can everyone see my slides?
Yep, thank you, Bowen. Go ahead.
Okay, thank you, Christine. Thank you so much for having me today. It is a great pleasure to present at the Prime Annual Symposium. So my name is Bowen Li. As Christine introduced, I'm currently a postdoc at MIT working with Professor Bob Langer and Ed Anderson, and I will join the Faculty of Pharmacy at UofT next January. So today I'm going to talk about my work in developing nanoparticulate delivery systems for RNA-based vaccines and therapy. And also we'll talk a little bit about my perspective about my future lab. So with an explosion of insights into the role of genetics in various diseases, nucleic acids confer tremendous flexibility and broader therapeutic utility than nearly all other classes of known drugs. They provide the unprecedented opportunity to make personalized medicine a greater reality in the clinic. They can take advantage of pre-existing cellular process to correct or reverse genetic abnormalities to treat diseases that were previously thought to be undruggable. Compared with DNA, RNA-based therapy has been receiving more attention in recent years because it affects a wide range of biological processes translationally without the risk of genetic integration. However, RNA also have its own unique characteristics that make it challenging to translate RNA to the clinic. For example, for mRNA, it is very large molecules with a molecular weight usually higher than 1,000 kilodalton. And meanwhile, it's highly unstable. It is vulnerable to nuclease once you apply it in vivo. And also it only works in the cytoplasm, which means that you have to deliver it across the cell membrane. So how can we deliver the RNA therapeutics inside the cells in vivo? So right now, the most popular platform for RNA delivery is the lipid nanoparticles. A classical ionizable lipid nanoparticle platform is composed of 4 components. The first component is the ionizable lipids, which is the key component because it plays a very important role in helping the mRNA to escape the endosome and release into the cytoplasm for translation. And also, it also plays a role to bind tightly and protect the mRNA from the degradation. And the second component is the structured haploids, such as DOP or DSPC. They also play a very important role in terms of help the lipid nanoparticle to fuse with the targeted cells. And the third component is pack anchor lipids. This lipid mainly used to stabilize the lipid nanoparticles and help to improve their solubility. And the fourth component is cholesterol, which plays an important role to help to improve the fluidity of the lipid membrane and help to stabilize the lipid nanoparticles. So a typical example of the use of lipid nanoparticles in the clinic is the mRNA vaccines. I believe that many of you have already been vaccinated using the mRNA vaccines. The mechanism of mRNA vaccine is basically pretty straightforward. So basically, the scientists figure out the sequence of mRNA that can encode the spike protein in the coronavirus. And then the mRNA sequence is loaded into the lipid nanoparticles. And once this lipid nanoparticles is injected into human, they will be able to allow the mRNA express into the spike protein that will later on induce the protective immune responses in patients. So the importance of lipid nanoparticles can never be more emphasized because it's really important, just as the title of the chemical engineering you'll say, without the lipid shells, there will be no mRNA vaccines for COVID-19. So my research interest is about how to build a suite of diverse lipid nanoparticles to enable next generation RNA vaccines and therapy. So we're interested in employing a combinatory library approach, which is driven by new chemistry to create new structures of lipids. And in this synthesis process, in the design process, we try to balance the novel tails with functional linkers as well as the core structures to achieve better effects. And then using the new lipids, we try to collect some in vivo data to establish structure-activity relationship, which can be used to further guide the lipid optimization. So now there's a problem, that is how to rapidly generate diverse ionizable lipids for use or for screening. So we recently developed a strategy called four-dimensional reaction. So in this reaction, we can quickly synthesize thousands of lipids overnight. And another unique point with this reaction is that different from the traditional synthesis process of lipids, our four-dimensional reaction includes 4 components. Okay, so which is different from the 2-component reaction used in the traditional lipid synthesis. And this 4-component reaction also does not use any toxic catalyst. It does not need for the solvent exchange, purification, or concentration. This allows us to quickly produce a large batch of lipids for scale up. So now I wanted to talk about how did we screen these lipids and how did we make use of these new lipids we developed. The first case I want to show is that we used our new lipid system to better improve the performance of mRNA vaccines. So I want to briefly introduce about the key components in the mRNA vaccine. So basically, the first component is the mRNA itself, which can encode the protein antigen. The second component is the nanoparticle, which serves as a delivery vehicle that can protect the mRNA from the nucleus degradation and can maximize the transcription of the mRNA in vivo. And the third component in mRNA vaccine that is usually ignored currently is the adjuvant. The adjuvant is a dangerous signal that can boost the non-specific immune response. So, but it is, although the adjuvant is widely used in traditional vaccines, but it is not usually aided in the mRNA vaccines. The main reason is because when you add adjuvants into the mRNA vaccines, it will negatively affect the mRNA translation to protein antigen. Therefore, in order to better improve the mRNA vaccines, we need to address the change of adjuvants. And we should think about how can we develop a new adjuvant that can amplify the immune responses, meanwhile does not affect the mRNA translation. So we have been thinking about, can we redesign our lipid nanoparticles to possess both high mRNA transfection efficiency as well as the adjuvanting properties? So as I mentioned that using the 4-dimensional reaction, we were able to design and quickly synthesize thousands of lipids. But how can we screen those lipids? Screening thousands of lipids can still be a tedious and really time-consuming work. So therefore, we designed a strategy called batch-based high-throughput screening. So we're using the Firefly surface mRNA, we were able to quickly screen out the best lipid tail A because in the 4-component reaction, we have 2 lipid tails, A and B. So what we did is basically we combine all of the lipids that share the same lipid tail A together, and we injected the same batch into the same mice. And in this way, we can quickly figure out which lipid tail A is the most promising one. And more in this batch of these testings, we picked out top performing lipid tail A, and then we combine the same. More in this pool, lipid pool, we combine all of the lipids that have the same lipid tail B and perform the same screening again. And more the top lipid tail A and B, we performed at the 3rd round of screening, batch-based screening. And in this way, we were able to figure out the top performing lipid tail A, B, as well as the linker. So afterwards, we were successfully narrowed down from thousands of lipids to 8 top performing lipid tail and the linkers. And at this moment, we start to optimize the hard group. So this is basically a step-by-step screening process. So in this process, in hard group optimization process, we not only care about the mRNA translation, we also want to incorporate some immunostimulatory functional groups into the lipid structures. So basically, what we expanded is that after we use the lipid nanoparticle to deliver the antigen mRNA, the lipid nanoparticle will be able to activate the dendritic cells and stimulate the dendritic cell to secrete the type 1 interferon, which will be able to promote the maturation of the dendritic cells and then further enhance the performance of the antigen mRNA in induced immune responses. So basically, we designed another big pool or big library of the hard groups. And this structure is mainly composed of cyclic lipid structures because we learned that the cyclic lipid structures tend to stimulate the immune responses more than the linear structure. So after we incorporate these diverse hard groups into the top performing lipid tail and the linkers structures, we were able to get another like hundreds of lipids. And then in this round, we just tested them in vitro 1 by 1 using the RF reporter monocytes, which will be able to tell us which lipids can stimulate the immune response. Basically, like if a lipid can stimulate the secretion of a type 1 interferon or activates this type 1 interferon pathway, we will be able to detect the signal from the reporter cells. So as you can see that in this heat map, we found that there are several lipids. As I circled, they show pretty high immune stimulatory function. This suggests that these lipids may be used as adjuvating lipids for the mRNA vaccine development. So as I mentioned that in order to further confirm whether a lipid is suitable for mRNA vaccine delivery, we have 2 standards. The first standard criteria is that we have measured that this lipid can lead to potent mRNA transfection. And secondly, this mRNA transfection will not be affected by its adjuvating effects. It is important that this lipid can also stimulate the immune responses better than the inert lipids. So as you can see here, so basically we further screened these lipids and we showed that there are several top performing lipids. They can still efficiently transfect the muscle after the intramuscular injection. So compared with MC3, so MC3 is basically an industry benchmark lipid that has already been used in clinic. So compared with the MC3, we can see that several of our lipids shown better transfection efficiency than the MC3 lipids. And we also showed that these lipids all have adjuvating effects, so which distinguish them from the conventional inert lipids that is currently used in the clinic, such as MC3. So this study shows that we were able to screen out lipids that can not only allow the mRNA translation, but also it can efficiently stimulate the immune system, which is very unique. And later on, we further studied the immunostimulatory effect of our new lipids in the case of mRNA delivery. And we found that the lipids we screen out can efficiently induce the antigen-specific T-cell responses. This property is very important because it suggests that we can use these lipid nanoparticles not only for the mRNA vaccines against infectious disease, but we can also probably develop cancer vaccines using these lipids. So we screen out a good lipid that is called number 143. This one shows the highest or the strongest ability to induce antigen-specific T-cell immune response. So therefore, we determined to move forward with the most potent lipids. And in this scenario, in this case, we try to demonstrate the application of our adjuvating lipid nanoparticles, which is also called, we call it AP, in further boosting the performance of COVID-19 mRNA vaccines. So in this case, we tested two model antigens. One is the wild type spike protein. Another is the RBD binding domain in the spike protein. It's called RBD. Both of them come from the SARS-CoV-2 virus, coronavirus. So basically, we encapsulated the two type of mRNA that encodes the SP and RBD respectively into our adjuvating lipid nanoparticles, and we performed two vaccination in the mice, and then collect the blood at different time points for ELISA test. We tested 3 different dosages in this case. As you can see that our lipid nanoparticles, or we call it AP mRNA vaccines, it can induce higher antibody titer than its counterpart, industry counterpart, Moderna mRNA 1273 vaccine at the same dose. So this results in suggest that the new immunofunctional or immunostimulatory lipids can not only deliver the mRNA or allow the mRNA to translate efficiently in vivo, it can also stimulate the immune responses without affecting the mRNA translation in vivo. So therefore, we basically confirm that the new lipid we screen out is a promising a new carrier for the mRNA vaccine delivery. And moving forward, we also found that when we change the chemistry of the lipids in the lipid nanoparticles, we can have the nanoparticles to target different organs. So this suggests that we can use our lipid nanoparticles to do some therapeutic work, such as do gene therapy. So our lab has received some funding support from a Translate Bio company, which is a company that aims to develop mRNA therapeutics for lung relevant diseases. For example, its pipeline, it has a pipeline called MRT 5005, which aims to deliver the mRNA that can encode the fully functional CFTR proteins for the treatment of cystic fibrosis. But unfortunately, this pipeline failed early this year because their lipid nanoparticle system is not potent enough, and not sufficient amount of protein were achieved in the lung. So therefore, we collaborated with Translate Bio to develop the more potent lipid nanoparticle delivery systems for the mRNA pulmonary delivery. So basically, we used the same high throughput screening four-dimensional high throughput screening and synthesis platform to synthesize 1,000 lipids and screen these lipids in the lung. And we successfully discovered or identified one lipid that is more potent in lung transfection compared with the benchmark lipids used by Translate Bio in its clinical trials. And this lipids can not only improve the transfection potency in the lung, it can also significantly reduce the toxicity because it is fully biodegradable. And then we want to test whether we can efficiently transfect the lung epithelial cells because the lung epithelial cells is the main target in the treatment of cystic fibrosis disease. So herein we use the genetically modified mice model. It's called AI14 mice model. So basically in this mice, it does not have in the normal status, this mice does not have any fluorescence. But once we deliver the Cray recombinant mRNA, the expression of the Cray recombinants will be able to cut off the stop sign and allow the T tomato fluorescence to be emitted. So we will be able to detect the the red fluorescence of T tomato signal. So what we did is that we encapsulate the Cray mRNA using our lipid nanoparticles, and we performed the 3 repeat dose of the intratracheal administration in the mice. And we found that after the intratracheal administration, our lipid nanoparticle can efficiently activate the lung epithelial cells. So as you can see in the red dots indicates the positive, the TD tomato positive cells. And the flow cytometry result further indicates that about 50% of the cell epithelial cells in the lung have been transfected. So this is a pretty high transfection efficiency, because people have found that if the minimum transfection percentage for therapeutic level is 24%. So which means that if we can transfect more than 24% of lung epithelial cells, we will be able to achieve therapeutic level. So moving forward, we further tested the gene editing effects in the lung epithelial cells using the same lipid nanoparticle. So this time we encapsulate the Cas9 mRNA as well as the gut RNA in our lipid nanoparticle system. And this time, again, we observed that about 35% of the lung epithelial cells in the large airway as well as the small airway has been successfully gene edited. So in a short summary, so basically we developed a throughput screening and synthesis platform for lipids. We can use this platform to quickly identify lipids with different function and for different purposes. For example, we can identify RNA nanoparticle system with dual delivery and adjuvant effects, and we can also identify the RNA nanoparticle system for non-viral gene editing. So basically, my lab will be focusing on developing the next generation advanced delivery platforms for RNA-based therapeutics and vaccines. So RNA is actually not limited to mRNA. It also contains siRNA, mRNA, and CRISPR-Cas9 systems. So it just provides a very powerful arsenal for us to use. So basically, in my lab, we will focus on to further use our high throughput synthesis and screening platform to identify new biomaterials that can first help this RNA therapeutics to overcome delivery barriers, including cell membrane, like mucus and blood-brain barrier. And secondly, we can further modulate the new biomaterials to have them deliver RNA to different organs or different cells. And thirdly, we want to integrate some new functions into the delivery vehicles by changing the structures of biomaterials. We hope to integrate such as stim activating or immune suppressor function into this new biomaterials so that we can achieve more functionalities for the treatment of a range of health diseases. So finally, I want to thank my advisors at MIT, Professor Bob Lange and Dan Anderson, as well as my PhD advisor, Xiaoyu Jiang at Cornell University. I'll be happy to answer any question if you have. Thank you.
Thank you, Bowen. We only have time for one quick question, if you don't mind. And so there's a question in the Q&A, which says, can you explain the lipid synthesis method further? Is it a one-pot reaction with all possible tails, cores, linkers, or is it split up into subgroups?
This is a great question. So basically, we do one-pot reaction with all possible tails and cores and linkers. So that's why we say that it can help us generate thousands of lipids. But in the screening step, we will further split these thousands of lipid nanoparticles, thousands of lipid structures into subgroups for screening.
Wonderful. Thanks very much, Bowen. And I'm going to ask Helen to maybe share a screen and we can get ready with the next presentation.
All right, my screen should be shared. Is that right?
Yep, perfect. Thanks, Helen. Go ahead.
All right. All right. Well, thank you everyone for coming to today's talk, and especially to Christine and Alan, I guess Christine and Christine for doing this Prime Symposium. I'm very excited to be here. I am a new professor in the Department of Chemistry, cross-appointed in Chemical Engineering and Applied Chemistry. I am also a AAAS IFTHEN ambassador, which serves to increase the public presence of women in media, specifically women in STEM. And so today I'll give you a very brief couple of slides about what I did in my postdoc and how that sort of motivated me to investigate these other research questions and what I'm doing right now in my lab. So if you take a look around you, we have all different types of electronics. When I was younger, we had electronics that were as heavy as me. In high school, I had a BlackBerry. Now we have iPhones, a 2-pound laptop, and maybe some of you are even wearing an iPhone, an Apple Watch, right? And what I think is in our future, these types of electronics are going to become even more seamless, functional, and information. There's already really nice examples of this, both in academia as well as industry. And we've seen that these new types of form factors can really improve how we interact with electronics. For instance, there's a beautiful example led by John Rogers and collaborators on using these wireless stickers effectively for monitoring ICU babies. And instead of having all these wires tugging on the very sensitive skin, we can just have a sticker allowing easier movement and skin quality not being damaged. And so we're looking at thinking about making these new types of electronics. At the core, in my opinion, would be the types of materials that need to be integrated. So we have skin, and skin is a source of inspiration for us because it's quite incredible. It's flexible, it's stretchable, it's self-healing, as well as biodegradable. And at the same time, it has these sensing functions of touch, temperature, and humidity. So can we create a material sort of like skin, having these mechanical properties, but in some ways electronic properties as well? And that's what we sought to do. And like many others, there's different ways of making electronic materials flexible and stretchable. You can think about making the substrate stretchable, put all your electronics on top, and then let it relax. So this is a really nice technique because we can directly get to stretchable electronics without re-engineering any of their existing techniques or materials. Similarly, with strain engineering, we can pre-strain, we can design devices that can accommodate for strain. So here you can see these sort of bridges that accommodate for stretching in different directions. And then lastly, we can think about making everything just intrinsically stretchable. And I'm particularly excited about this area because it comes down to a materials challenge and thinking about the molecular design of materials. Indeed, this is great because you don't have as much integration issues, but their electronic performance needs to match that of the inorganic counterparts. And there's been quite a lot of progress, particularly for materials that are stretchable and even self-healing. And the two types of materials that we're particularly interested in are semiconductors and conductors, because they're much more difficult to create rather than materials that are just inert. And while there are good examples of this, one field that is lacking is making these materials also biodegradable. So now we have sort of a paradigm where we need to create a material that's stretchable, electron conducting, as well as biodegradable. And this allows us to start having really new types of devices that we can think about making. So for example, here's a really nice example from the collaboration between Paige Fox and Jianan Bao, where they thought about how to make a wireless pressure sensor so that you can implant it post surgery for a vessel anastomosis. So vessel anastomosis is when you take two vessels that were disconnected and reconnected it. And this happens in bypass surgeries as well as other surgeries. The key in this type of surgery is after the surgery, it's unclear whether your seal was sufficient. you may detect that by a drop in blood pressure, but did that happen because you had a failure in the surgery, or you just had an open heart surgery, so your blood pressure decreased? So thinking about creating a localized pressure sensor that degrades over time will allow us to monitor the healing process of this surgery. And these type of examples are really enabled by thinking about materials with different types of properties. Like I mentioned, we have electron and ion conductivity, mechanical softness and stretchability, as well as degradability and recyclability. So getting a material that allows us to have all three of properties is really a molecular design issue. And for semiconductors and conductors, this becomes very difficult to do because many of the molecular design rules that make something electron conducting is not what was needed for making something degradable, and vice versa with mechanical properties. That said, this is possible. This is some work that I did as a postdoc in Janan Bao's group at Stanford University. We thought about using both polymer chemistry and polymer physics in order to engineer a material that has all three of these mechanical properties. It has the right morphology, it shows the right electronic performance. You can see we made field effect transistors that upon stretching from 0 to 100 and even release, the performance of the material stayed the same versus a material without the type of polymer physics that we engineered into the system will decrease in electronic performance over orders of magnitude. We can tell that these materials are degradable by monitoring it by UV vis. And in an initial study, we see that these materials are also biocompatible with cells. So, working on these types of projects, I had a few questions in starting my own group. And one really big question is thinking about how to integrate these types of electronics with the body. How do we use it for precision medicine? And to me, I think the answer is both polymer science and engineering, because now we have two handles to really tune how to create new types of materials. And in my group, we think about using polymer chemistry and for creating soft electronics. And really the key is using self-assembly processes. And the three areas in my group that we look at are using functional bottle brush polymers for modulus match electronics, thinking about how this particular type of architecture allows us to maintain electron conductivity, but also mechanical softness. Thinking about self-assembly to build electronics bottom up and ideally in vivo. And lastly, reimagining what types of monomers we can use to build up our conjugated polymers. Because today's talk is for Prime, I wanted to give a bit of insight on our bottom-up bioelectronics, because there's quite a bit of precision involved in these types of devices. And so sort of thinking back about what Ray said this morning about multi-specific drugs that sort of work by induced proximity, I like to think about multivalency and how it is important for selective binding of many receptors to their target molecule. We see that material that doesn't have multivalent interactions will not behave as well as a material with multivalent interactions. And you can think about that like the birds, if you ever gone hiking, or Velcro as well. And this has been implemented in drug delivery and detection service applications as well. There's a very nice globular example where they create mannose, but in a multivalent motif that showed really high increase in inhibition relative to its non-multivalent counterparts. Similarly, we can think about looking at DNA architectures to have the precise location of aptamers to get really selective binding. I think Wendy today, one of the Prime Fellows, talked about DNA as an architecture, and this is an example of that, where they place, they try to mimic the distribution of what the dengue virus looks like, and so they created a star-like architecture with aptamers at one of these apexes, and upon binding, binding of the aptamer, the star opens up and then leads to fluorescence. And you can see that it is very specific to the dengue virus, and even against adenovirus, there's no background fluorescence showing here. And so thinking about this sort of problem, I wanted to, recall what I did as an undergraduate at Berkeley with Ron Zuckerman. And we thought about using peptoids as a platform for detection and multivalent interactions. We worked on showing that you can use these biomimetic materials to bind proteins in a multivalent manner, observe it by fluorescence. And you can even integrate it with carbon nanotubes. So now we're starting to step in closer towards integrating biomimetic materials with electronic properties. And these are really good initial advances, and there's still some techniques that we need to grow on that. And that's what I want to work on here. Can we think about using sequence control for precise nanostructures, and can they self-assemble in such a way to have multivalent binding events? And now I'm thinking about, induced proximity for future examples as well. And so peptoids are these materials that feature particular types of sequence between hydrophilic and hydrophobic, and they can self-assemble in different ways. One example is at the air-water interface, and you can even incorporate a peptide as loops. And these materials self-assemble into 2D sheets, and how we design the sequence of the material and where we put the loops, we can even display molecules, peptides at the surface. And this can be observed by atomic force microscopy here. These peptoids are synthesized by solid phase synthesis, which is very straightforward. It's very similar to peptides, and we have a robotic synthesizer to synthesize these materials on scale. And the key with peptoids is now I can think about a lot of different things to integrate. Besides the hydrophobic, hydrophilic domains, we can think about incorporating stimuli responsive materials and really components that can introduce electron conductance into the materials through traditional conjugated polymers, post-polymerization, as well as asymmetric semiconductors for pre-polymerization instead of post-polymerization, and as well as more metal coordination. And one area we're looking at is using diacetylenes, which can be topochemically polymerized. And these types of materials are similar to acetylene, a very conductive polymer that can be generated in solid state. So thinking about how to incorporate these materials into the sheet, we may be able to create materials that can display peptides and other motifs, but also have electron conductance. Besides, using organic compounds, we can also look at inorganic compounds to induce to create metals and other types of materials here. Skip this slide. And then with that, this is an idea of what these materials will look like. We have these free floating 2D sheets that sort of look like tissue paper floating in solution. We can use a variety of material science technique to observe how they self-assemble and what kind of mechanical properties that they have. And so with that, I'd like to thank my group, especially Jan Babby and Abigail Clapperton, who are two graduate students that work with me on the peptoid subgroup, and all the various fundings that have supported me through the years. And I'm happy to take any questions.
Thank you so much, Helen. Sorry, I was having a hard time unmuting myself. Thank you for that great presentation. So I'll just ask if there are any questions for Helen, just please add those to the Q&A. I know we've got a couple of questions for Bowen that have actually come in after the fact. And so perhaps we can ask the individuals that are asking those questions to reach out to Bowen directly by email. Any questions for Helen? Please go ahead. And if not, then maybe in the interest of time and staying on schedule, we'll move on to the next speaker. Thank you, Helen.
Mm-hmm.
So our next speaker is Rafael Montenegro Burke. He's an assistant professor in the Department of Molecular Genetics, and his team focuses on the development of strategies for novel metabolite characterization and discovery to expand our view of conserved metabolic pathways and their impact on cell function in health and disease. Take it away, Rafa.
Sorry, I was muted.
Oh, there we go. Perfect.
Can you see me? Sorry about that.
Yep.
Well, thank you for the invitation. It's exciting to speak today at the PRIME Symposium. So I'll be telling you today about how we use metabolomics to go from beyond biomarkers and towards deciphering unknown biology. And I'll be telling you today about two particular projects, one that we worked during my postdoc time and some of the collaborations that we started to do here in some of those projects as well. But first, what is metabolomics? As you know, genomics is a discipline in which the complete set of DNA within cells or an organism is analyzed. Similarly, metabolomics is a discipline where the metabolites within cells or an organism is analyzed. And what we consider metabolites are essentially small molecules involved in metabolism. And those can be lipids, amino acids, carbohydrates, sterols, and many other types of molecules. And why are we interested in metabolites or metabolomics? As you know, metabolomics is at the bottom of the omics cascade from genomics and proteomics. And being at the bottom, it's really close to the phenotype. So it's a really good readout of what the genome and what the proteome did. And it gives us a really good overall picture of what is happening within a cell. The approach that we use is called on-target metabolomics. And what it allows us to do is in a very unbiased way, profile and characterize tens of thousands of different metabolites in a single experiment. Now, this gives a really good picture of what the phenotype of what is happening, at a molecular level. So our targeted metabolomics workflow looks like this. We can analyze cells, tissues, biological fluids, and we use liquid chromatography mass spectrometry to separate and detect the different molecules. Then we do some data processing, some statistics, which give us an idea of what the metabolome output looks like. We use these statistics to then prioritize certain dysregulated metabolites to then, in combination with a fragmentation spectra, called here MSMS experiments, we can compare with databases and identify these metabolites. Once we've identified these metabolites, usually in the field of metabolomics, they're called biomarkers. But in our lab, we're interested in going beyond that to help us understand biology a little bit better. And I'll be telling you today, like I said, about two projects. In the first one, I'll tell you about a metabolite activity, how metabolites regulate inflammation. And this was in the context of IBD. And the second project is how a medulloblastoma rewires metabolism and how we can exploit some of those processes to discover metabolic vulnerabilities. First, let me start with the IBD project. Why we're interested in IBD? IBD affects a really large population. For example, in Canada alone, it's around 270,000 people. And this disease does not affect only adults, as around 7,000 of these patients are children or under the age of 18. The number of patients suffering from IBD is going to increase, and it is expected that around 1% of the population by 2030 will have some sort of IBD. Now, the exact reason or what causes IBD is not completely understood, but there have been several factors that have been identified, such as genetics, environment, and diet. In terms of genetics, around 1 in 5 patients has another family that also suffers from IBD. And in terms of the environment and the diet, this seems to be, in Western diet, seems to be more people are suffering from IBD. So this was really the motivation for us. As well, that changes in diet seems to be a good treatment to relieve some of the symptoms from IBD. So when we think about what is the diet, we think about lipids, amino acids, sugars. Those are all metabolites. So we thought of using our technology that allows us to, in an unbiased way, profile thousands of metabolites and try to understand what are those metabolites that could be actually contributing to the phenotype of IBD and influencing inflammation. So our experimental design was we use an animal model, have wild type and the IBD group. We have collected fecal, cecal, and colon content, and we used our normal metabolomics workflow. And this gave us a list of dysregulated metabolites that in the field are usually known as biomarkers. However, we're interested in getting a better understanding of what the function is of these metabolites. And when we look at the metabolites in the different regions, we observe a really broad range of chemistries, from amino acids to sugars, lipids, vitamins, sterols. In many cases, some of them downregulated here in yellow, some of them in purple, and it's really, really broad. And I don't expect you to look at all of them. But what we're interested here, remember, is what is the function or what are these metabolites doing in the context of inflammation? Are these metabolites influencing the inflammation levels? Can this tip the balance towards senescence or towards inflammation? So what we did is we screened these metabolites for their metabolite, for the activity. And essentially the question we asked was, can these metabolites induce inflammation or increase the inflammation levels, or can they reduce inflammation? And for that, we use a broad range of different cell culture assays using both mouse and human macrophages, and we look at canonical markers of inflammation. We use this to determine which metabolites had an interesting effect on this phenotype of inflammation. And we discovered this metabolite, phytosphingosine, that I'm showing you here, that is downregulated in IBD. That means it's found in lower concentrations in the colon of mice that are suffering from IBD. And the effect that we observed in all these cell culture-based assays was that it was anti-inflammatory. This obviously made it really, really promising to us to pursue further and move on and try to test this in vivo. We then did this in a mouse model, and we observed that actually reduces inflammation in vivo models as well. As I'm showing you here, the relative mRNA levels of MCP1, a canonical inflammatory marker, where in gray is the vehicle, in red is the TNBS, which is the name of the model for inflammation. And then in blue is TNBS treated with phytosphingosine with oral gavage, where we see a drop of these MCP1 mRNA levels. So taking this together, we know that now metabolites actively are contributing to the inflammation in IBD. And this is really powerful if you think that we can use these metabolites to our advantage. We can use the metabolites in a purposeful way to actually modulate the phenotype. And some research, some of the studies project in my lab involve understanding what the metabolism and what are the pathways that phytosphingosine uses to regulate inflammation. And this happens not only in humans, but also not only in mice, but also in human macrophages as well. So it gives us really, really good opportunities to try to understand what are the targets for us to use. Now, I want to switch gears and talk about a project that we established, that we're doing in a collaboration, and that is looking at the metabolic rewiring medulloblastoma. As you know, metabolic rewiring is a hallmark in cancer, right? We did this, we're doing this collaboration with Sheila Sim lab at McMaster, and a lot of this work has been done with a very talented postdoc, Will Green, that I'm showing you here in the picture. So we know medulloblastoma is a very heterogeneous disease. And for this particular project, we focus on group 3 medulloblastoma. These groups are divided based on some of the expression of the genes, and they have different characteristics. Group 3, for example, has a very poor prognosis, and the proposed cell of origin is neural stem cells. So what we're doing is just using in vitro modeling to determine what are the molecular pathways, what are the metabolic pathways that are contributing to the development of medulloblastomas, and actually can we use our workflows to understand what are the metabolic differences here between natural neural stem cells and medulloblastoma to identify potential metabolic vulnerabilities. So we detected several metabolic changes, and I want to focus just on some of the lipid classes that we observed. For example, I'm showing here the different lipid classes, and this is the trend for all of the lipid species that belong to that class. And what we observe is here in neural stem cells, we observe a significant levels of the hydroceramides compared to the medulloblastoma, as well as the sphingomyelins that I'm showing you here. But I thought today I wanted to use this opportunity to be a little bit more unconventional and show you how rich the data is in metabolomics and how we can extract more information from different layers from this metabolomics data. And for that, I want you to direct your attention to a group here of triglycerides, where in reality, we don't observe at a first glance like really broad or significant changes. As you know, triglycerides are composed of this glycerol backbone right here, and 3 fatty acids are attached to that triglyceride. The nomenclature that we use for fatty acids in lipidomics is composed by 2 numbers. The first number is the length of the chain. In this case, for this species here at the bottom, 16.2, 16 represents the number of carbons. And then the number 2 is the number of double bonds, or the level of saturation. So I want to first start, I want to tell you about what this level of saturation of the double bond numbers means, or what it's actually doing in medulloblastoma with triglycerides, for example. So what I'm showing you here in this plot, I know there are many bars, but it can be simplified. Here at the bottom, we see around 40 different triglyceride species, and they're ranked based on the number of double bonds. The saturated lipid species that have 0 or only 1 double bond are here on the left. And the unsaturated levels increase as we move to the right here, between 4, 5, and 6 double bonds for all these triglycerides. On the y-axis, these are the normalized levels from medulloblastoma, for example, to neural stem cells. That's why neural stem cells marked here in red are all at 1. And when we look at this plot, look at this sort of wavy trend, we observe that we see higher abundance of these triglycerides for the medulloblastoma around 0 and 1. So medulloblastoma seems to be using more of the saturated fatty acids for these type of lipids. And we observe not a lot of unsaturated lipids are being used for making these triglycerides in medulloblastoma compared to the cell of origin neural stem cells. Now you're thinking, what does this double bond actually mean or represent? Or is it really biologically important? And we're really excited about this data, particularly after Will sent me a slide with some interesting data on Saturday. And what I'm showing you here is using a drug that is involved in some of these lipid pathways right here, which unfortunately I cannot reveal. We see how specific and what the vulnerability it is for medulloblastoma to be treated with this drug compared to neural stem cells. So what I want to show you here essentially is that even though we don't observe significant changes in the levels of triglycerides as a whole, when we start looking more in detail what the different composition of these lipid species is, we can identify certain vulnerabilities that are not that apparent at a first glance. Now, in summary, I hope I was able to show you how we use on-target metabolomics and how it provides valuable insight into cellular processes, and how we can go from beyond biomarkers and try to understand more about the biology. Also, how we can actually use on-target metabolomics to identify metabolites that are actually playing active roles in the modulation of phenotypes. And in the case of medulloblastoma, how we can map the different metabolic pathways and the rewiring steps and processes, and that helps us to identify potential diseases and vulnerabilities. I'd like to acknowledge the team, the people that do the hard work in the lab and the funding as well. And I'm an assistant professor cross-appointed in the molecular department, in the Department of Molecular Genetics, but also at the Donnelly Center, which I'm showing you a picture right here. Ruby, thank you for your attention. I'll be happy to take any questions.
Wonderful. Thank you so much, Rafael. What a great presentation. So amazing to see all these new assistant professors at UofT In the interest of time, we're just going to move on to the next presentation. We're actually a little bit behind in our schedule now. But if anybody has questions for Rafael, please include them in the Q&A and he can answer the questions there. Of course, you can also reach out directly to him. So thank you very much. And our next presenter is Chris Lawson, an assistant professor in the Department of Chemical Engineering and Applied Chemistry. Welcome, Chris.
Thank you very much. I'm just getting my slides up here. Hopefully you guys can see these.
Perfect.
Great. Okay, well, thank you so much for the invitation and for the introduction, Professor Allen. And so my research is focused on understanding and engineering microbial communities or microbiomes, largely in industrial and environmental applications, actually. But a lot of the work that we're doing is quite fundamental on microbiome engineering. And so I think there's lots of interesting or useful information for precision medicine as well. And so in today's talk, I'll focus on some of the applications we're working on in the lab, and then also highlight how this might be applicable to precision medicine. And certainly we're very interested in seeking partnerships with people in the prime community. So if people are interested in collaborating, happy to chat further after the talk. Okay, so broadly, we're interested in this overarching question of developing systematic approaches for microbiome engineering. And so when we think about microbial systems, we're not only having to think about how an organism's genotype translates into a phenotype, which is typical of a single organism, obviously, but we're also interested in understanding how individual phenotypes between cells and local interactions between cells translate to emergent phenotypes at the ecosystem scale. And so our lab is generally interested in defining or developing tools to quantify and manipulate metabolism. And so, we're interested in thinking about if we have a genetic change here in an individual organism, how that translates to the ecosystem scale, and likewise, how ecosystem scale environmental changes might alter the phenotype of an individual organism. And so our lab is interested in the general goal of converting wastes and biomass to valuable chemicals. And you can see here, this is done using very complicated microbiomes, much like you'd expect to see in the human gut or other areas of the human microbiome. And so the main questions we're really interested in are thinking about, can we identify, which microbes are performing a particular metabolism in this complicated food web of interactions, how we might be able to precisely alter microbiome composition. So in this food web here I'm showing, we have complex organic polymers being degraded, in this case into methane and short chain fatty acids, but maybe we don't want this one that produces methane. So how would we selectively remove it from the microbiome, or maybe reroute flux toward the product that we have of interest? And thirdly, can we genetically manipulate specific organisms within a microbial community? And so here, this microbe I'm showing you here, maybe it produces fatty acids, but could we get this microbe instead to produce alkanes or different metabolites such as methyl ketones? So these are the overarching questions our lab is generally interested in. And of course, this doesn't just apply to these digester microbiomes, which we're interested in, but of course, the human microbiome and other areas in precision medicine as well. And so to illustrate some of the things our lab is working on, I thought I'd go over a simple example where we're trying to convert plant biomass into volatile fatty acids. And so for this project, we actually draw a lot of our inspiration from the rumen microbiome. And so here's a picture of a cow, and it turns out that a cow is actually quite a sophisticated bioreactor. It has a milling operation here at the front end of the cow that sends plant biomass into the rumen, where anaerobic microbes and microbial communities convert that biomass, or the sugar content in that biomass, into volatile fatty acids via anaerobic fermentation pathways. And the cow can do this the cow microbiome can do this very, very efficiently. About 75% of the energy content in the biomass is actually retained as volatile fatty acids. And so what our lab is interested in doing is thinking about how we might be able to repurpose this as an industrial scale bioreactor. So instead of this cow model, can we repurpose this or get these microbial processes happening in some type of industrial scale tank? Okay, so to get this started, to first assemble the communities that we're interested in developing, what our lab did is we went out and we started enriching different microbial communities from the rumen as well as these anaerobic digesters. So essentially what this is going out, collecting some fecal samples from the cow or the goats and some sludge samples from digesters. And then we inoculate it with grass anaerobically in what are known as Hungate tubes. And we can propagate these every 3 or 4 days or so for months. And what we end up doing is we end up selecting for rich communities that can convert the plant biomass into different products that we're interested in. And we've separated here the bacterial enrichments from the fungal enrichments, just using different antibiotic selection mechanisms. And we're really interested actually in these aerobic fungi because they're well known to have very good biomass degrading capabilities. And so what our idea now is to further isolate these organisms from these enrichments, and this is something we're well on our way to doing, and then recombining them in some intelligent way to create synthetic microbiomes. And so one possibility here might be to have the anaerobic fungi, which are, as I mentioned, specialists in lignocellulose degradation, convert lignocellulose to different fermentation products, and then different microbes or bacteria from the bacterial enrichments, taking those fermentation products to volatile fatty acids. So this is just one example of how this might work. Of course, there's a lot of diversity within these enrichment cultures and lots of possibilities for how this picture should work. And so to better inform how to build these synthetic microbiomes, what we first did is we took a deep dive on the microbiome composition and function of these bacterial and fungal enrichments. And so I'll present a little bit of data about that and then tell you how we're now approaching our synthetic microbiome construction. And so here again are just the different enrichments that we had. So we had these 3 enrichments from the cow, the goat, and the digester on the bacterial side. And we were also able to get enrichment cultures growing from the fungal enrichment cultures from the cow and the goat. And so what I'm showing you here is just a plot of how much biomass is being degraded into soluble products, so just the biomass that is lost. And what you can see is that, in the green bars here, the fungi were very efficient at degrading or solubilizing biomass. Up to 60% of the biomass is solubilized in the products. The bacteria, not quite as good, but also able to funnel biomass into particular products. And when we look at the metabolites, the one metabolite we were particularly interested in recovering was butyrate. So that's a C4 fatty acid. And as you can see here, the brown bars is showing the bacterial enrichments, and the green bars is showing the fungal enrichments. Really, the bacteria that were the ones that were capable of degrading the biomass into butyrate. And we also, of course, measured a lot of different other fermentation products. And we recovered a lot of the intermediates that might be, along the path to butyrate. In particular, in the green here, this is showing formate lactate and acetate, which was really highly produced by the fungi, characteristic of a mixed acid fermentation pathway. And we also had other products such as propionate and acetate by the bacterial community, which shows that not all of our feed that we're interested in converting to butyrate is actually going there. So there's opportunity to improve this yield. And so there's lots of questions that we can ask here, but one of the main ones, of course, that we were interested in was which microbes and pathways were actually responsible for producing this butyrate, that were a response that we're interested in. And so in order to do this, what we did is we performed metagenomic sequencing on both all these bacterial enrichments because they were the ones producing butyrate. And we also profiled their gene expression using transcriptomics. And so what I'm showing you here is just some bioinformatic work we did. And so what we did, we were able to bin out hundreds of different bacterial genomes from these enrichment cultures, and then map back our DNA and RNA reads to profile their abundance and activity in the community. So what you can see here in the light bars is just the RNA, overall RNA expression of that genome, and in red is the DNA abundance. And one of the striking things that we saw was that across both the digester and the cow enrichments, I'm just showing you 2 example plots here, was that these lactospiraceae organisms were really highly abundant and also very active. And so each genome you see here is representative of maybe a different species of lactospiraceae. So we had quite a high diversity of these lactospiraceae in our community, and they were very active. And so, of course, one of the next questions is, what metabolic pathways were they expressing? and are they responsible for butyrate production? And so this is just a really overview summary of the different pathways that we were able to reconstruct in across all the different genomes. And so this heat map is just showing you the different genomes we had versus pathway. And so, as to be expected when you're feeding a sugar substrate to a community, we had a lot of different hexose and pentose metabolism, namely glycolysis and the pentose phosphate pathway, which would take different sugar monomers to fatty acids, or sorry, pardon me, to different fermentation intermediates. And so here you can see what we're able to do is we can look at the gene expression of individual organisms and particular reactions in that organism, and we can get a sense to what they might be doing based on which genes they're really highly expressing. And this works quite well actually for anaerobic communities. And it turns out when we looked across the genomes for pathways involved in butyrate production, they are very, very selective for these lactose-peraceae. And so this is this reverse beta oxidation pathway, which can take 2 molecules of acetyl-CoA and elongate them into butyrate. And if you keep going around the cycle, you can actually get longer fatty acids as well. And so here, if you look at the heat map, you can see that these pathways for the reverse beta oxidation were very enriched in the, or expressed, pardon me, by the lactose-peraceae organisms. And so to put it all together, what we've been doing is we've been using this type of analysis to understand what role or ecological niche each microbe might have in the community. And so we've been able to, as I said, identify the role of different lactose-peraceae or propose some roles for these lactose-peraceae involved in butyrate production, as well as other microbes in the community that may take these sugars to different intermediates of interest. And we've also been able to identify particular competing interactions. And so here, for example, we also noticed that there was a lot of sulfate-reducing bacteria that were enriched in our community that are taking intermediates that typically funnel into butyrate and instead respiring them to CO2 using sulfate as an electron acceptor. And so the question is, with these enrichments, can we improve upon this by building synthetic microbiomes? And so, you can imagine that there's lots of different synthetic microbiomes you could propose, but it's not really clear which one is optimal. So as we could take a pool of these lactose-peraceae and just have them converting the glucans into butyrate, for example, but maybe the biomass conversion might not be as high. We could take some of the better bacterial communities that were able to utilize the more complex polymers and couple them with these lactose-peraceae, or we could take some of the fungi that we observe are very good at degrading biomass as well and coupling them with some of these butyrate producing organisms. So there's lots of different, pathways and combinations of microbes you can envision. And of course, there's a wide variety of different environmental conditions that you could then select these communities under. And so what this really calls for then is a systematic approach for thinking about designing, building, and testing these different synthetic microbiomes. And so what we've done now is we've taken those enrichment cultures and we've applied or performed some high throughput isolations. And so we've isolated about 420 different functional members of the microbial community on the bacterial side. And we've also isolated a number of the fungi from our fungal enrichments. And so we have this large library now of different isolates that we're thinking about combining in a systematic manner to really optimize the production of, in our case, butyrate, or really generally any other function you'd be interested in. And so the way we are starting to do this, so we're just building a large facility in our lab to do this using liquid handling in an anaerobic setting. But we're thinking about, you could take different, you could combine different consortia under different conditions, and maybe some of these consortia are expressing different enzymes as well. You could build them using our facility, which is a liquid handling robot under anaerobic conditions, coupled to different screening approaches. You could test those different consortia, media, and enzyme combinations using mass spec, for example. And then what you can do is you can take that data, so all your different design combinations and their resulting performance, in our case, the butyrate titer or yield, and you can use that data to train different machine learning algorithms that would not only learn the relationship between some of these design variables and their performance, but also enable you to then recommend what design criteria might lead to improved in our case, butyrate titers for functional outputs that you're interested in. And so this really then enables an automated and data-driven design, build, test, learn cycle for optimizing different microbiome engineering properties that you're interested in. And so this is something that our lab now is just starting. We just opened up September, so we're just getting this all going in the lab. And in addition to this, so we're not just interested in making butyrate, but we're also interested in a more specific control of metabolism. So instead of making, the C4 compounds, could we control the chain length of the medium chain fatty acid we're interested in? And so that might require engineering some thioesterase in that pathway. But we're also interested in taking these medium chain fatty acids and making them into alcohols, alkanes, methyl ketones. And so, maybe this should require adding some different chemistry to these microbiomes that's not naturally present in them. And so in order to do this, this really requires the engineering then of non-model organisms. And one of the key things that has been a bottleneck for engineering microbes, non-model organisms, is really restriction modification systems. So we have a project on the go right now to engineer around these native defense systems common in especially anaerobic bacteria such as Clostridium. So in our anaerobic bacteria, a lot of their DNA is methylated. And so if you introduce plasmids into the genome or into the cells, they get chewed up. And so we're developing approaches that allow us to identify the methylase activities and then methylate our plasmids prior to entering transforming our cells. And this has been shown to really increase transformation efficiency. And the other thing we're just starting to get interested in is also this in situ metagenomic engineering. And so in a number of cases, not only are we interested in engineering the individual microbes, but we'd like to engineer them in situ, if you will. And so here, what we're working on is engineering a donor cell that could then transfer a plasmid that we've already corrected for with this methylome, identifying their methylome via conjugation into a native cell that's residing in the microbiome, and then have that propagate through the system. And so these are two general areas that we're really interested in from a microbiome engineering perspective as well. And so, of course, these approaches are not only relevant for anaerobic digestion, but we think they're also very relevant for precision medicine as well. And so there's just been a really recent publication that's come out looking at how metabolic engineering has been applied to cancer therapeutics. And so in this example, they're engineering gut bacteria to recycle tumor waste in the form of ammonia that then increases local arginine concentrations for T cells and makes them more effective at killing tumor cells. And so there's lots of applications for the work that we're doing. We think in problem areas like the tumor microenvironment. And so we're interested to chat with folks that would like to collaborate on some of these microbiome engineering pursuits. There's a lot of commonalities with them. And so with that, I'd like to thank you for listening to my talk. Here's my team that just started up in September, so really excited to have them on board. And I'm also very thankful for some of the collaborators that we still have at the Joint Bioenergy Institute and Oak Ridge National Lab. And I forgot our logo, but also some of the funding that we've received from NSERC. So with that, I'll take any questions. Thank you.
Fantastic. Thank you, Chris. I love the picture and the logo. Amazing.
Thanks.
So we will move on just in the interest of time. We're running late today, so my apologies for that. If anybody has questions for Chris, please include them in the Q&A panel and he can respond there. You can also reach out directly. Just a huge thank you to all four of you, really, Bowen, Helen, Rafael, Chris, thank you so much for sharing your research with us today. And my goodness, is the future ever bright at the University of Toronto with all of you on board. So we will now